Neuromuscular junction. This animation will demonstrate the events taking place at the neuromuscular junction. The production and release of acetylcholine creates action potentials that lead to skeletal muscle contraction. Pharmacological agents that affect this process, as well as disease states that occur when this process is disrupted, will also be reviewed. Here, you can see a snapshot of the neuromuscular junction, which shows the axons of a motor neuron and the muscle fiber. The neuromuscular junction serves as the synapse between a motor neuron and the muscle fiber it activates. Now, let us take a closer look at the neuromuscular junction. We'll start by looking within the cytoplasm of the motor neuron axon terminal. Pay close attention to the labeled elements, such as acetyl-CoA, choline molecules, and choline acetyltransferase, as well as the areas surrounding the nerve terminal. Acetyl-CoA produced by mitochondria combines with choline molecules to form acetylcholine via an enzyme known as choline acetyltransferase. Once acetylcholine is formed, it is packaged into synaptic vesicles. Action potentials arrive at the presynaptic neuron terminal, causing voltage-gated calcium channels to open. This allows calcium ions to flow from the extracellular fluid into the cytosol of the presynaptic neuron. Influx of calcium causes acetylcholine-containing vesicles to dock and fuse with the presynaptic neuron cell membrane. This results in an emptying of acetylcholine into the synaptic cleft, a process known as exocytosis. Now let's focus our attention on the events taking place within the synaptic cleft. Acetylcholine diffuses into the synaptic cleft and binds to nicotinic receptors, located on acetylcholine-gated ion channels, which are on the postsynaptic membrane of the muscle cell. Once bound, acetylcholine causes these channels to open, allowing sodium ions to flow in and potassium ions to flow out of the muscle cytosol. Due to the electrochemical gradient across this membrane, more sodium moves in than potassium out. Influx of sodium produces a local depolarization of the motor end plate, known as an end plate potential. The action of acetylcholine is terminated by two processes. First, acetylcholine is rapidly degraded by acetylcholinesterase enzymes to produce choline and acetate. The remaining acetylcholine diffuses away from the neuromuscular junction. The choline molecules created by the degradation of acetylcholine are then recycled. They undergo reuptake back into the presynaptic terminal, where they will be used to synthesize new acetylcholine molecules. We will now review conditions related to the neuromuscular junction. Lambert-Eaton syndrome is an autoimmune disorder of neuromuscular transmission. In this syndrome, autoantibodies block calcium channels on the presynaptic membrane of the axon terminal. This prevents calcium ions from moving into the synaptic cell. Therefore, acetylcholine cannot be released into the synaptic cleft. Myasthenia gravis is another autoimmune disorder that affects the neuromuscular junction. In this syndrome, autoantibodies attach to postsynaptic membrane nicotinic receptors. This prevents acetylcholine from binding to nicotinic receptors. Therefore, sodium cannot enter and depolarization cannot occur. Pharmacology. The mechanism of action of nicotinic agonists, like succinylcholine, involves a persistent depolarization of the neuromuscular junction. The persistence of this depolarization occurs because succinylcholine mimics the effect of acetylcholine, but cannot be hydrolyzed by acetylcholinesterase. This leads to continued depolarization of the postsynaptic membrane and subsequent desensitization of this membrane to additional releases of acetylcholine. Pharmacology of acetylcholinesterase inhibitors. Acetylcholinesterase inhibitors, such as neostigmine, are used in the treatment of myasthenia gravis and Lambert-Eaton syndrome. We will first review the normal physiology and then the action of acetylcholinesterase inhibitors. Again, after acetylcholine enters the synaptic cleft, it binds to nicotinic receptors, allowing sodium ions in and potassium ions out. Acetylcholine is then rapidly degraded by acetylcholinesterase enzymes to produce choline and acetate. Acetylcholinesterase inhibitors prevent the degradation of acetylcholine by acetylcholinesterase. This increases both the amount of acetylcholine and the duration of time that it remains within the synaptic cleft of the neuromuscular junction. Pharmacology of nicotinic receptor antagonists. In this animation, the normal interaction between acetylcholine and nicotinic receptors is shown on the right. On the left, nicotinic receptor antagonists are seen preventing acetylcholine from binding to nicotinic receptors. Thus, depolarization cannot take place. Nicotinic receptor antagonists, such as atracurium, inhibit nicotinic receptors on the postsynaptic membrane. This prevents acetylcholine from binding at these sites. Nicotinic receptor antagonists are mainly used for peripheral muscle paralysis during surgery. Action potential causing muscle contraction. We will now show how the generation of an action potential leads to skeletal muscle contraction. As mentioned before, the influx of sodium ions across the postsynaptic membrane causes local depolarization. The magnitude of depolarization is referred to as the end plate potential. Skeletal muscle membranes in the synaptic region do not have voltage-gated sodium channels. Therefore, action potentials cannot occur in this region. However, the end plate potential can spread to adjacent areas, causing depolarization of muscle membranes adjacent to the end plate, where voltage-gated sodium channels are present. Their opening causes the initiation of action potentials across the surface of the skeletal muscle cell. The action potential travels down the T-tubule and activates dihydropyridine receptors. These receptors pull the junctional foot processes to open ryanodine calcium channels in the sarcoplasmic reticulum. This causes calcium ions to leave the terminal cisterna of the sarcoplasmic reticulum. Cross-bridge cycling occurs when calcium ions attach to troponin, causing tropomyosin to move and expose the attachment sites for myosin cross-bridges. It is important to understand that the binding of ATP to the myosin cross-bridge contributes to its movement. With muscle contraction, the I-band and H-band of sarcomeres are shortened. Here, the end result of the interaction between acetylcholine and nicotinic receptors within the neuromuscular junction can be seen causing muscle contraction.